Hi. So in this series, we are going to discuss about the DPPs of PW 2.0 batch. This is the DPP number 4. We have already discussed about DPP 1, 2, and 3 in the previous videos. So let's start with DPP 4 of inheritance biology, that is genetics, unit number 8 from CSIR point of view. So let's go to the first question. Which one of the following situations could be termed as intergenic suppression of a mutation in a coding sequence? Okay, so in that case, we are getting a mutation in a gene and another mutation happening in another gene, but these are complementary to each other. So a nonsense mutation in the codon leading to lack of interaction. So if it is lack of interaction, that couldn't be survived by another mutation. The AMBER mutation in the codon recognized by altered anticodon in tRNA. Yeah. So if another AMBER mutation, that is stop AMBER means example of stop codon. So if the stop codon is detected by a altered tRNA anticodon. So that could be a intergenic mutation, that two of the gene having mutation. And read through of the stop codon. This is also non-specific, very non-specific mutation. And frame shift mutation skipping and otherwise stop codon. So if frame shift mutation skipping the stop codon, to survive from this mutation, we have to we have to introduce another mutation in the same gene. So this could be an example of intragenic but not intergenic. So this is not the answer. Answer is question answer number two. No, option number two. Next, a single base pair mutation occurs in strand, and mutant strand is sequence CATT-ACCG, while its complementary strand has the sequence GA GTT-ATGGC. So let's match this G C with that C with G. Uh, if we see the let's see this is the main complementary strand. That this is the here is mutation. So the main strand or the previous strand is this. So G T T A A T G T T A T G G C. This is the previous strand. So the complementary should be C A A T A C C G. What is there? C A T T A C C G. So this T is complementary. This T is mutated. There should be an A, but this mutated into a T. So the mutation changed is adenine to thymine, A to T mutation. Next, point mutation in which there is deletion and addition of one base pair is termed as there is there is deletion or addition in one base pair. So if this is addition of deletion occurs in one base pair, that could change the entire coding region, entire coding sequence. So that could be termed as frame shift mutation. The frame shift mutation is induced by normally acridines. UV rays are very important for thymine dimerization or such kind of things. If If substitution is the only possible type of mutation, then which technique would be no more useful for detecting them? Substitution. Substitution could not be detected by DNA sequencing can detect, PCR amplification couldn't, database searching can detect again, restriction fragment analysis can detect again because this is also a site specific. But PCR amplification couldn't detect a substitution mutation. If mutation changes codon in such a way that there is no effect on functioning and sequence of protein, if mutation in changes in such a way there is no effect on sequencing sequence of protein, this type of mutation is called missense mutation. This is where the mutation is occurred but no changes in protein is known as known as missense. Sorry, this is silent mutation. This is mutation occurred but no no any particular notification, no any particular symptoms. So this could be termed as silent mutation, not mutation. Next, bacterial operon contain three structural gene A, B, and C in the same order. If polar mutation occurs in the gene B, then the effect of protein would be observed in all protein A, B, C, only B and C, and only B. Complete loss of all protein. See, if a bacterial operon have three genes, that means A, we can say B, and we can say C. So A is already synthesized, the mRNA or mRNA for A is already synthesized and protein is also synthesizing by the ribosome simultaneously. But if mutation occurs in B, the B stops and options protein number C also stops. So we cannot find the B and C protein in the bacteria. So only affecting B and C. Consider the following DNA sequence A T G G C C. Insertion of G between third and fourth. Okay, one, two, three, fourth. So this segment, this site has another G sequence lead to film-shift mutation and non-functional protein synthesis. Which of intragenic suppressor mutation will have the minimum effect on functioning of the protein? The inserts of nucleotide between 5 and 6 position. See, if we insert any one nucleotide and we have already inserted one nucleotide, then the film shift is still there. So we cannot insert there uh, to suppress that mutation. So we have to delete one nucleotide. That could be option 3 and 4. There is deletion occurring. So let's say about 5 and 6 and 11 and 12. 1, 2, 3, 4, and this one is 5, this one is 5th, and this G is 6th. So if we delete one mutation, delete one uh, particular nucleotide from 5 and 6 that are also G, and the nearby from the G insertion we did previously. So that would be most favorable because the sequence will remain same. And if we delete 11th and 12th, then we can see something about from this side, and that could uh, changes the main sequence. So option C will be the more correct. Next, an insertion of single nucleotide in coding region of gene leads to frame shift mutation. Yeah. Result as a result is formation of non-functional protein. Under certain conditions, second suppression mutation is in another gene may result into formation of functional protein. How suppression mutation can do this? Okay, so this is a French mutation, but this could be suppressed by another gene, another mutation in another gene. Let's see the options. There is insertional mutation in the gene of tRNA anticodon such as it is able to interact with 4 nucleotide codon. Interesting. Mutation in gene of ribosome leading to frame shift over transcript. Ribosome has no any such function in reading. Mutation in gene whose product bulge out extra nucleotide. Mutation in gene whose product bulge out extra nucleotide, but that is not a separate gene mutation. That should be an intragenic, not intergenic. So this is also not the answer. And another mutation reverse the original mutation. This is also an intragenic. Intragenic, not an intergenic. So option one is the only option we can pick. Spontaneity of mutation means spontaneity of mutation. The mutation is in absence of exogenous mutagen, that could be an answer. Mutation directly proportion to presence of mutagen, that is not spontaneous. Mutation in immunoprimary right, mutation inversely proportional to presence of mutagen. No. Option A. Spontaneity is in absence of exogenous mutation. We didn't give any particular mutagen, but the mutation occurs. That is spontaneous. Mutation in gene X leads to lethality in a haploid organism. Which one of the following is best suited to analyze the function of gene X? Pleiotropic mutation, temperature sensitive couldn't be an answer. Recessive mutation, this is a haploid organism, so recessive mutation is not the answer. Mutants with low penetrance. Penetrance doesn't says about the function of gene. So pleiotropy is the only option. Aspartic acid is specified by codon GAU and GAC. After mutation, aspartate is changed to alanine represented by GCX, where X may be A, U, G, C. The transversion of the mutation could only be done with reactive oxygen species. The nature of mutation is considered as so. In that case, the nature of mutation is obviously transversion because GA something was the codon for GAC or GAU was the codon for aspartic acid, and it is converted into GCX. So A to C transition, A to C mutation is there. So A to C, that is purine to pyrimidine, and purine to pyrimidine mutation is known as transversion. So this is a transversion. Question number C. A gene encoding tRNA undergoes a mutational event in its anticodon region and region that enables it to recognize a mutant nonsense codon and permit completion of translation. Such mutation is known as so this mutation on the T cell which can find a mutant codon. So this could be a mm, Nonsense suppression could be a possible reverse. This is not a reversion. Reversion is there is a mutation, and we did the second mutation, and the previous main wild type sequences uh, regained. But in this is not a silent mutation also. This is not a neutral mutation also. This is a perfect example of nonsense suppression. Because in silent mutation we get some this kind of mutation, that is the previous one was GAC, and the mutated one is happened as GAU. Then both are coding for aspartic acid. So this kind of mutations are silent mutation. The neutral mutation is the similar type of thing. A silent mutation and neutral mutation are very much similar. Next question: hypothetical gene encodes a protein with the following amino acid sequence: phenylalanine, proline, threonine, alanine, valine, arginine, serine. A mutation in a single nucleotide alters the amino acid phenylalanine, leucine, 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 leucine. A second single nucleotide mutation occurs in the same gene, restoring back the amino acid sequence to the original. The following statement were made regarding the nature of the location of the first mutation and that the intergenic suppression mutation. Okay, so we can see that phenylalanine is same. Then proline, from the proline, all the nucleotide change. This is a case of frame shift mutation. So the first mutation is deleting in the second codon. First mutation is a deletion in the second codon. Yeah, that could a possible reason. First mutation is the insertion of second codon. That's all. That also a possible. But see, if we have a insertion, then we have to consider that the number of amino acid will remain same. But if there is a deletion, the number of amino acid will decrease in one. So there is a decrease. So option B is not correct. There couldn't be an insertion in first mutation. And intergenic suppression mutation is an insertion of second codon. Yeah, that's a true. Not a deletion. It's an insertion to get the previous sequence. So option A and C is correct. A and C is correct. Next question: the effect of nonsense mutation could be nullified by reversion as well as suppression. Okay. Which of the following process will help distinguish between two kinds of revertant? So, complementation, transgenesis, test for allelism, and recombination. Recombination is the most perfect way to do such things, because if we do the reversion, the original sequence is back. But if we do suppression, the original uh, form is not back, but we are suppressing that by doing another mutation in another gene. If we recombinant these two things, the recombinant will make a the recombinant we will get could be possibly detached or crossover that to particular gene. So in that case, we can found recessive mutant. So we can say option 4 is the correct answer. And that's it for today. Thank you.